Adenosine triphosphate, commonly known as ATP, is often called the molecular currency of cellular energy. It is the primary energy carrier in all living organisms, from the smallest bacteria to the largest animals. Every cellular process that requires energy, whether it's muscle contraction, nerve impulse transmission, or protein synthesis, relies on ATP. Understanding the structure of this remarkable molecule is essential to comprehending how cells store and release energy for life's processes. ATP is composed of 3 distinct structural components that work together to store and transfer energy. The first component is adenine, which is a nitrogenous base containing nitrogen atoms in its ring structure. The second component is ribose, a 5-carbon sugar molecule that forms the structural backbone. The third component consists of 3 phosphate groups, which are the key to ATP's energy-storing capabilities. These 3 parts are chemically bonded together to form the complete ATP molecule, with each component playing a critical role in the molecule's function. Adenine is a purine base, meaning it has a double-ring structure made up of carbon and nitrogen atoms. This double-ring configuration distinguishes purines from pyrimidines, which have only a single ring. Adenine attaches to the ribose sugar at a specific location called the 1' position. This connection point is crucial for maintaining the molecule's stability. Beyond providing structural support, adenine serves as a molecular recognition site, allowing enzymes and other proteins to identify and interact with ATP specifically. This recognition is essential for the molecule's role in countless biochemical reactions. Ribose is a pentose sugar, meaning it contains exactly 5 carbon atoms arranged in a ring structure. This sugar molecule serves as the central connector in ATP, linking the adenine base to the chain of phosphate groups. The carbon atoms in ribose are numbered with prime symbols to distinguish them from the atoms in adenine. The 2' and 3' positions on ribose contain hydroxyl groups, which are oxygen and hydrogen combinations. These hydroxyl groups are important for the molecule's chemical properties. The 5' carbon is where the phosphate groups attach, creating the energy-rich portion of the molecule. The 3 phosphate groups in ATP are designated by Greek letters, alpha, beta, and gamma. The alpha phosphate is positioned closest to the ribose sugar, attached directly to the 5' carbon. The beta phosphate sits in the middle of the chain, connecting alpha and gamma. The gamma phosphate is the terminal, or outermost, phosphate group. These 3 phosphates are connected in a linear chain extending away from the ribose. The bonds between these phosphate groups are critical because they store the chemical energy that makes ATP such an effective energy carrier in cells. The bonds connecting the phosphate groups are called Phosphoanhydride Bonds, and they are considered high-energy bonds. Specifically, the bonds between the alpha and beta phosphates and between the beta and gamma phosphates store significant amounts of chemical energy. When the terminal gamma phosphate is removed through a process called hydrolysis, approximately 30.5 kilojoules per mole of energy is released. This energy release occurs because the phosphate groups are negatively charged and naturally repel each other, creating an unstable, high-energy configuration. Breaking these bonds relieves this tension and releases energy that cells can harness to perform work, making ATP the universal energy currency of life.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.